Okay, we're going to take up today, we have here the seminal fluid analysis. So our seminal fluid analysis here is divided into your macroscopic examination or simply your physical exam. Then you could also have here your microscopic examination. Okay, so upon receipt of the specimen laboratory, you need to take note of the following macroscopic parameters. Okay, the first one, we have here the appearance. So try to take note of the color. Okay, then we have also the volume. The volume is being done here by transferring your entire volume of your specimen into your graduated cylinder that would allow you to measure the volume. Okay, the odor, you try to take note of that. The pH. So normally, the pH of our normal seminal fluid would be 7.2 to 8. You can measure the pH by your pH meter or can also have your reagent strip. You can also use your even your litmus paper. Then the viscosity can be identified here by transferring a sample of your seminal fluid in your pipette and allowing it to flow at the tip of your pipette. Okay, then we have here your microscopic examination. So before you can perform your microscopic examination, make sure that your seminal fluid has been liquefied already. Liquefaction takes approximately 30 to 60 minutes after the collection process. And then again, you could not proceed with your microscopic examination not unless your specimen has been liquefied already. Okay, for the microscopic examination, first, you should perform here the sperm count. Okay, for the sperm count, we'll be using here your RBC pipette. You can also use your WBC pipette. And then your diluting fluid in the form of your sodium bicarbonate plus or formaldehyde. You can also use your NSS. You can also use here your cold water as your diluting fluid. Okay, then you just need to have also here your Neubauer counting chamber. Okay, so perform when you are computing here for the sperm count. First, you should perform here your sperm concentration um, procedure or the counting. The sperm concentration count here, or the sperm, you call this one as your sperm concentration. Okay, so basically you can you can get that one by doing your, okay, the Neubauer counting chamber and then try to count and then try to um, use here the standard na formula for the computation. Just like when you're doing your other routine na mga cell count. So again, it's up to you if you use here the RBC pipette. If you're using the RBC pipette, then just use your standard formula for the computation of that. And if you're using here WBC pipette, then you try to use also here the standard formula when you are doing your WBC count. And whatever the result you get from that, that's your sperm concentration. In order for you to get your sperm count, okay, you need to multiply your sperm concentration by the total volume of your specimen. Okay, another method here we'll be doing here would be your sperm motility evaluation. So the sperm motility evaluation here is not only for the speed of the motility of your sperm, but as well as their direction. So sperm, ano ito, it should be the motility, the speed, and the direction. So it should be progressive and forward motility dapat. Okay, procedure for this one. Okay, so just be needing here concave na slide. So 1 drop of your sperm, liquefy the seminal fluid on your concave na slide. And then after that one, try to cover with your cover slip. You try to seal the edge of that with a petroleum jelly. And then try to observe in your 10 HPO. How are you going to present your sperm motility here? You try to present that one in terms of the percentage. So there are 10 HPO. So in every field, how many are motile? How are you going to compute for the motility? So you need to count how many of those sperms are in your 1 HPF field. And out of that one, how many are motile? Like for example, if you have 100 na sperm cell in 1 HPO and there are only 50 are motile or moving, okay, that's 50%. Okay, then you go to the next na HPO field until you come up with a 10. And the percentage of that for your 10 HPO field, you try to divide, that become your sperm motility. Okay, the normal sperm motility should eventually be at least 50% within 1 hour of collection process. Okay, the next one we have here your sperm viability. The sperm viability actually measures or identify how many of your sperm cells which are not moving, which are actually dead, or how many of those sperm cells which are not moving, we are still alive. Okay, so minsan kasi some are not moving but they are still alive, so they are still viable. However, meron namang hindi gumagalaw na patay na talaga. But we are after of here na hindi gumagalaw but they are still alive. Okay, that's what you are measuring here with your sperm viability. Okay, procedure for this one. So you have your seminal fluid. You try to put that one in your slide. Try to make a smear, a thin smear. Then after that one, you try to stain. So the stain used here for the sperm viability is your using nicrosin stain. Then air dry. After that one, you try to evaluate under your OIO. And try to count here your alive. Okay, so you're going to count at least 100 na sperm cells and then identify here or try to compute how many are viable in such a way that they are unstained. Usually they are bluish white. All the other sperms are purple, orange. So those are dead. Ang mga alive natin na mga cells here are those unstained, usually bluish white in color. Okay, that's the number of your alive cells or your unstained cell over 100 times 100 to become your percent viability. The normal viability should approximately be 75% dapat. Okay, another one, we have here the sperm morphology evaluation. Okay, there are two sperm morphology evaluation. One, we have here the routine morphology evaluation where we are just observing here for the morphology of the head, the tail, or even the neck part. Another one is your Kreuzer's three criteria wherein it try to measure. Really, meron siyang sariling measurement, kaya nga three criteria siya. So for you to observe here for the sperm morphology, first you need to make a thin smear, air dry, and try to stain. Okay, for the staining, so we have that one. You try to fix that one. Wash with your phenol bicarbonate. Okay, to remove here your mucus. And then try to stain with your base of fusin. Counter stain with your methylene blue. And then try to observe under the OIO for each of the morphology. So you're going to count here or you're going to evaluate 200 sperm cells. And how many of those are abnormal? So paano natin masabing abnormal? Like, if meron siyang abnormality in the head, like double head, pinhead, or may abnormality in the tail, like double-tailed and all. Like, pag, for example, if you're doing here your routine criteria na morphology evaluation, pag may abnormality sa head, kahit normal ang tail niya, so still counted pa rin siya as abnormal. So all you need to do is just count here or evaluate out of your 200 na sperm evaluated, how many are abnormal. Okay, then multiply by 100 that become here your percent sperm morphology. Again, for the routine criteria, so normal morphology natin should at least be 30%. And for the Kreutzer's three criteria, so at least be 13%. But most likely, routinely, we are just having that routine criteria or routine morphology evaluation lang naman ginagawa natin. Okay, so that's it for our sperm analysis. Okay, thank you.